This is not just a podcast. This is your turning point. Every minute you revise today saves you from panic tomorrow. So focus, breathe, and study like. Your future depends on it, because it does. Welcome to Biomania Revision Podcast. Put on your headphones, close your eyes, and listen carefully. This is not entertainment. This is training for your exam. All right, target is set. We are drilling down into NCERT Class 11, Chapter 12, Respiration in Plants. This is where biology and chemistry collide.
It's where students lose the most marks. Calculations, complex names, carbon counting, it's a minefield.
But not today. Today, we're not just reading lines. We are decoding the entire mechanism. So sit up straight, no slouching.
Total focus is required. Respiration in plants is the absolute backbone of plant physiology. If you don't understand how energy is made, you can't understand growth. Period. Let's attack the definitions first.
Starting with the most basic definition, the first point of confusion, breathing versus respiration. People think they're the same. They are not. Give them the correct definition.
Respiration is the breaking of the CC bonds of complex compounds through oxidation within the cells to release energy.
Listen to those keywords, oxidation and energy release.
Exactly. And the cell can't just use that raw energy. It's too chaotic. It has to be trapped in the form of ATP.
That point is crucial. So CC bonds are breaking. Who's CC bonds? We call those respiratory substrates. And the cell has a priority list. What gets burned first?
Glucose. Always glucose. Carbohydrates are the preferred substrate.
What if the cell is starving?
Then, and only then, it might turn to fats or proteins, and in some very specific conditions, even organic acids. But you must remember, pure proteins or fats are never used directly. They have to be modified first.
And now for a direct question that shows up as a statement in exams. Do plants breathe? Do they have lungs? The answer is a hard no.
Plants have no specialized respiratory organs. Animals have lungs, they have gills. Plants have nothing like that. Why? 3 reasons. Memorize all 3. Reason number 1, independence. Look at a tree. The roots are deep underground, the leaves are 50 feet in the air, the root needs oxygen. It's not going to ask the leaf for it.
So every part takes care of itself.
Every single plant part, root, stem, leaf handles its own gas exchange. The transport of gases from one part to another is almost non-existent. Each part is self-sufficient.
Okay, that's reason 1. Reason number 2.
Low demand. This is where students get confused. They think, living thing, high demand. Wrong! Plants aren't running marathons. Their muscles aren't contracting.
Their energy requirement is slow and steady.
Exactly. So the rate of respiration is incredibly low compared to an animal's. The demand for gas exchange is minimal.
What about during photosynthesis? Gas exchange is huge then.
But think about it. During photosynthesis, the cell is producing its own oxygen. It doesn't need to take it from the outside. The supply is internal. It's only when photosynthesis stops that it needs external oxygen, and by then the demand is very low.
Right. Reason number 3, diffusion distance. Picture a thick tree trunk. You're thinking, how does air get to the very center?
The answer is it doesn't have to. The center of the tree, the pith, is made of dead cells, and dead cells do not respire.
So only the living cells need oxygen.
Precisely. And the living cells are located just near the surface, right under the bark. In leaves, the cells are arranged in thin layers, so the gas doesn't have to travel far.
And the tissue itself helps.
Yes. The parenchyma tissue is loosely packed. It creates these interconnected air spaces that give the gas an easy pathway.
So the structures are clear. Stomata in the leaves, lenticels in the stems. Those are the openings. Now the chemical equation. Burn this into your memory.
The general equation for aerobic respiration. C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus energy.
But there's a warning here. This isn't combustion. If you just burn glucose in a lab, all that energy escapes as heat. The cell would be incinerated.
And a cell doesn't want heat. It wants chemical energy. It wants ATP.
Which is why this process is not a single step. It's a series of slow, stepwise reactions. Each step is controlled by a specific enzyme.
Right. It's all about control. Releasing the energy in small, manageable packets that can be converted into ATP. No explosions.
Okay, the process begins now. Glucose has entered the cell, phase 1, glycolysis.
Write down its other name, the EMP pathway, for Emden, Meyerhoff, and Parnas, the 3 scientists who mapped it out.
Location is critical. Where does glycolysis happen?
In the cytoplasm or cytosol?
And the most important question of all, does it need oxygen?
Absolutely not. Glycolysis is an anaerobic process. It's common to both aerobes and anaerobes. It is the universal pathway.
From bacteria to humans, glycolysis is the same.
The exact same. Now, the starting material, in plants, glucose isn't usually floating around freely. The transport form is sucrose.
So first you have to break down the sucrose. The enzyme is invertase.
Invertase breaks sucrose into glucose and fructose. Both of these can then enter the glycolysis pathway.
Okay, focus up. 10 steps. We're dissecting every single one. There are two main phases. First, we spend ATP the investment phase. Then we make ATP the payoff phase.
Step 1. Activation. Glucose, a 6-carbon molecule, is stable. To make it react, you have to make it unstable. We do that by adding a phosphate group.
The reaction, glucose plus ADP, yields glucose 6-phosphate plus ADP.
And the enzyme.
Hexokinase. Hexo for the 6-carbon sugar. Kinase for an enzyme that transfers a phosphate from ATP requires Mg++ as a cofactor.
Note it down. 1 ATP has just been consumed.
Step 2. Isomerization. Glucose 6-phosphate rearranges into its isomer, fructose 6-phosphate. The formula is the same, just a structural change.
Step 3. Another investment. Fructose 6-phosphate still isn't reactive enough. We're adding another phosphate, this time at the other end.
The reaction. Fructose 6-phosphate plus ATP yields fructose 1part6 bisphosphate.
Notice the name. It's bisphosphate, not diphosphate. That means the phosphates are on 2 different carbons.
And the enzyme here is phosphofructokinase, or PFK. This is the pacemaker, the rate-limiting step of the whole process.
And crucially, the second ATP has been consumed. The cell is now 2 ATP in debt.
Step 4. Splitting. Now, the 6-carbon fructose 1-calorie-6 bisphosphate is cleaved right down the middle.
It splits into 2 3-carbon molecules. One is PGAL, or 3-phosphoglyceraldehyde. The other is DHAP, dihydroxyacetone phosphate. They are isomers of each other.
But the pathway continues with PGAL.
Yes. DHAP can't proceed directly, so an enzyme, triose phosphate isomerase, quickly converts the DHAP into another molecule of PGAL.
So what's the result of the splitting phase?
The result is 2 molecules of PGL. From this point on, every single reaction happens twice. You must multiply everything by 2. Do not forget the multiplier.
Now the payoff phase begins. Time to make some energy. Step 5. Well, technically step 6 in the full sequence.
This is the critical step, oxidation and phosphorylation. PGAL is converted to BPGA, or 1,4,3-bisphosphoglycerate.
2 things happen here. First, PGAL is oxidized. That means hydrogen is removed.
And who's the hydrogen carrier? NAD plus NAD plus gets reduced to form NADH plus H. Since we have 2 PGL molecules.
We get 2 NADH.
Correct. And notice an inorganic phosphate from the cytoplasm is added here, not from ATP. This is key.
Step 7. The first ATP generation. The high-energy BPGA donates one of its phosphates directly to ADP.
BPGA becomes 3-PGA, or 3-phosphoglyceric acid, and we get ATP. Since there are 2 molecules, we get 2 ATP.
And the name of this process is substrate-level phosphorylation. Why?
Because the phosphate came directly from the substrate. There was no electron transport chain involved. It was a direct transfer.
Okay, step 8. Isomerization. The phosphate on 3-PGA moves from the 3rd carbon to the 2nd. It becomes 2-PGA.
Step 9. Dehydration. A water molecule is removed from 2-PGA. The enzyme is enolase. This creates a very unstable, high-energy compound.
PEP, phosphoryl pyruvate.
Exactly. And now, for the grand finale, step 10, the final ATP generation.
PEP is converted into pyruvic acid. It's highly unstable, so it gives up its phosphate group to ADP.
Forming ATP, again, because we started with 2 PPPs, we get 2 more ATP. This is also substrate-level phosphorylation. The enzyme is pyruvate kinase.
All right, stop. Pen down. Let's do the math.
The investment. We used 1 ATP in step 1 and 1 in step 3. Total cost, 2 ATP.
Payoff. We made 2 ATP in step 7 and 2 ATP in step 10. Total produced 4 ATP.
So the net gain of ATP is 4 produced minus 2 consumed. That equals
2 ATP, a net gain of 2 ATP.
And what else did we get? We got 2 NADH back in step 6, and the final product.
2 molecules of pyruvic acid, each with 3 carbons.
Check the carbon balance. We started with 1 6-carbon glucose. We ended with 2 3-carbon pyruvic acids. No carbon has been lost. CO2 is not released in glycolysis. Remember that.
So now the big question. What happens to this pyruvic acid? Its fate is decided by one thing.
Oxygen. Is oxygen available or not?
Case A, no oxygen, anaerobic conditions.
Yeah.
The cell goes into fermentation.
Oxygen is present, aerobic conditions. Pyruvic acid moves on to the Krebs cycle. Let's deal with fermentation first.
Right. Fermentation happens in yeast, some bacteria, and even our own muscles during heavy exercise.
Yeah.
And the location is still the cytoplasm.
The main goal here isn't to make more ATP. The goal is to regenerate NAD+.
Why is that so important?
Think back to glycolysis. Step 6 needs NAD+ to proceed. If all the NAD+ gets converted to NADH and there's no oxygen to take the electrons, the cell runs out of NAD+.
And glycolysis stops. The cell dies.
So fermentation is a rescue mission to free up NAD plus Electron. Type 1, alcoholic fermentation. Example, yeast.
This happens in 2 steps. Step 1, pyruvic acid is decarboxylated. A CO2 molecule is removed.
The enzyme is pyruvic acid decarboxylase. The product is acetaldehyde.
Step 2, the acetaldehyde is then reduced to form ethanol. And this is where the NADH is used. It gives up its hydrogen, becoming NAD+ again.
The enzyme is alcohol dehydrogenase. End products, ethyl alcohol and CO2.
Type 2, lactic acid fermentation, found in bacteria like lactobacillus and in our muscles.
This one's a single step. Pyruvic acid is directly reduced by NADH to form lactic acid. The enzyme is lactate dehydrogenase.
Critical point, no CO2 is released here. Pyruvic acid has 3 carbons. Lactic acid also has 3 carbons. No carbon loss.
Now, for the exam, you need to know the drawbacks. Why is fermentation so inefficient?
1, energy yield. It's pathetic. Less than 7% of the energy in glucose is released. Most of it is still locked up in the alcohol or acid.
2, net ATP. The only ATP you get is the 2 from glycolysis. Fermentation itself produces 0 new ATP.
And 3rd, the products are toxic.
Highly toxic. Yeast cells produce alcohol, but once the concentration hits about 13%, it poisons them and they die. That's why natural fermentation for wine and beer stops around that level.
Okay, now let's shift the scenario. Oxygen is available. The plant cell switches to aerobic mode.
Pyruvic acid doesn't stay in the cytoplasm. It is actively transported into the mitochondria. To be specific, into the mitochondrial matrix.
And this next step is the bridge between glycolysis and the Krebs cycle. It's called the link reaction, or the gateway reaction.
Right. Pyruvate, a 3-carbon molecule, can't enter the Krebs cycle directly. The cycle needs a 2-carbon unit to start.
So we have to remove 1 carbon as CO2.
The process is called oxidative decarboxylation. Decarboxylation means removing CO2. Oxidative means removing hydrogen.
Let's hear the reaction.
Pyruvic acid plus coenzyme A plus NAD plus yields acyl-CoA plus CO2 plus NADH plus H cup.
The enzyme complex here is a big one, the pyruvate dehydrogenase complex. It's not a single enzyme, but a group of them, and it requires Mg++ ions to function.
Now update your calculations. We had 2 pyruvate molecules from 1 glucose.
But this reaction happens twice.
Meaning we produce 2 acetyl-CoA, we release 2 CO2. This is the first time carbon is lost in aerobic respiration, and we trap energy in the form of 2 NADH.
And now with acetyl-CoA formed, we enter the mate event, the Krebs cycle.
Also known as the tricarboxylic acid cycle, or TCA cycle, or the citric acid cycle.
Named after Hans Krebs. Location, mitochondrial matrix. Now, you need full concentration. We're tracking carbon numbers step by step.
The cycle kicks off with a condensation reaction. Step 1.
Acetyl-CoA, which is a 2-carbon unit, combines with a 4-carbon molecule already in the matrix called oxaloacetic acid, or OAA.
2C plus 4C equals 6C. Water is used, coenzyme A is released, and the product is the 6-carbon molecule citric acid. The enzyme is citrate synthase.
Step 2. Citrate is isomerized into isocitrate. Still 6 carbons.
Step 3. This is a major step, the first decarboxylation. Isocitrate 6C loses a molecule of CO2.
And it's also oxidized. NAD+ is reduced to NADH.
The product is alpha-ketobutyric acid, and the carbon count. 6 minus 1 is 5. This is the only 5-carbon intermediate in the entire cycle. Mark that.
Step 4. The second decarboxylation, alpha-ketogluteric acid, 5C, is also oxidized and decarboxylated.
Another CO2 is released, another NADH is formed, coenzyme A is added back in. The product is succinyl-CoA.
And the carbon count. 5 minus 1 is 4. From this point on, all intermediates will be 4-carbon compounds.
Step 5. Substrate-level phosphorylation. The bond holding coenzyme A to succinate is a high-energy bond. When it breaks,
that energy is used to convert GDP into GTP, which is energetically equivalent to ATP. Succinyl-CoA becomes succinic acid.
So for our purposes, we can say 1 ATP is directly produced here.
Step 6, oxidation of succinate. Succinic acid is oxidized to form fumaric acid, but there's a catch here.
The oxidizing agent is an NAD+, dears. It's FAD, flavin adenine dinucleotide. FAD is reduced to FADH2.
And the enzyme for this step, succinate dehydrogenase, is special. Where is it located?
It's not free in the matrix like the others. It is embedded in the inner mitochondrial membrane. This is the same enzyme that will later function as complex 2 of the electron transport system. That's a classic exam question.
Step 7, hydration. Water is added to fumarate, converting it to malic acid. Still 4 carbons.
Step 8, the final oxidation. Malic acid is oxidized to regenerate our starting molecule,
Oxaloacetic acid, OAA.
And in the process, one last NAD plus is reduced to NADH. The cycle is complete. The OAA is now ready to pick up another acetyl-CoA.
Okay, time for accounting. Let's tally the yield for one turn of the Krebs cycle from one acetyl-CoA.
We got 3 NADH, 1 FADH2, 1 ATP from GTP, and we released 2 molecules of CO2.
But remember, one glucose molecule produces 2 acetyl-CoA. So the cycle turns twice, multiply everything by 2.
The total yield per glucose from the Krebs cycle is 6 NADH, 2 FADH2, and 2 ATP.
And 4 molecules of CO2.
Now, check your total CO2 count. 0 from glycolysis, 2 from the link reaction, 4 from the Krebs cycle. That's 6 CO2 in total.
The 6 carbons from the original glucose molecule have all been oxidized and released as CO2. The glucose is completely broken down, but where's the energy?
The vast majority of the energy is still locked away. It's in the high-energy electrons being carried by NADH and FADH2. To cash them in, we have to go to the bank.
And the bank is the electron transport system.
The ETS, and oxidative phosphorylation. Location shifts now. We are on the inner mitochondrial membrane.
That membrane is folded into cristae to maximize the surface area for these reactions. And here we find 5 protein complexes. You have to know them in order.
First, the concept. NADH and FADH2 will drop off their high-energy electrons. These electrons will pass down a chain of carriers, like a relay race.
As the electrons move downhill in terms of energy, they release that energy in small bursts.
And that energy is used to do work, specifically to pump protons, H+ ions, from the matrix across the inner membrane into the inner membrane space. This creates a powerful gradient.
Let's break down the complexes. Complex 1.
NADH dehydrogenase. It does exactly what its name says. It takes electrons from NADH, oxidizing it back to NAD plus Weiss. Those electrons are then passed to a mobile carrier called ubiquinone.
And as this happens, complex II pumps protons into the intermembrane space.
Complex 2, succinate dehydrogenase. This is where FADH2 drops off its electrons, getting oxidized back to FAD.
These electrons also go to ubiquinone, but there's a key difference.
Complex 2 does not pump protons. This is why FADH2 yields less energy than NADH. It enters the chain at a lower energy level.
Okay, so now ubiquinone, a mobile carrier within the membrane, has the electrons. It carries them too.
Complex 2, the cytochrome BC1 complex. It accepts electrons from ubiquinone, passes them on, and in the process pumps more protons.
From complex 3, the electrons are picked up by another mobile carrier.
Cytochrome C. This is a small protein on the outer surface of the inner membrane. It acts like a shuttle, physically moving to carry electrons from complex 3 to complex 4.
Complex 4, the end of the line. Its name is cytochrome C oxidase.
It contains cytochrome A and cytochrome A3. And a key detail for exams, it also contains 2 copper centers.
And what does complex 4V do with the electrons?
It passes them to the final electron acceptor. This is the whole reason you breathe. It's oxygen.
Oxygen takes the electrons, combines with protons from the matrix, and forms water.
Exactly. $12 O2Ls plus 2H plus 2E right RO H2O1. If there's no oxygen, complex 4 gets stuck with electrons. The whole chain backs up. Everything stops. ATP synthesis halts, and you die.
So far, all we've done is pump protons. We've created a massive proton gradient. High concentration in the intermembrane space, low concentration in the matrix.
And those protons want to flow back down their concentration gradient, but the membrane is impermeable to them. They can only get back through one specific gate.
Complex V.
Also known as ATP synthase. It has 2 parts. The F0 part is embedded in the membrane and forms a channel for the protons to pass through.
And the F1 part is a headpiece that sticks out into the matrix. This is where the ATP is actually made.
The process is called chemiosmosis. As protons rush through the F0 channel, they cause the F1 headpiece to spin. This physical rotation provides the energy to slam ADP and inorganic phosphate together to make ATP.
Now, for the conversion rates, you must memorize.
For every 1 NADH that is oxidized, enough protons are pumped to generate 3 ATP.
And for every 1 FADH2.
Only 2 ATP because it skipped complex I and pumped fewer protons.
Okay, final segment, the respiratory balance sheet. Let's calculate the total theoretical ATP yield per glucose molecule.
This is based on a few assumptions, of course, that the pathway is sequential and no intermediates are pulled out for other uses.
Let's count it up. Stage 1, glycolysis.
You get 2 ATP directly by substrate-level phosphorylation, and you get 2 NADH.
Those 2 NADH go to the ETS and each produce 3 ATP, so that's another 6 ATP. Total from glycolysis is 8 ATP.
Correct. Stage 2, the link reaction.
No direct ATP.
Okay.
You get 2 NADH.
And those 2 NADH are worth 6 ATP in the ETS.
Okay. Stage 3, the
Yeah.
Krebs cycle. You get 2 ATP directly from GTP. You get 6 NADH, which are worth 18 ATP. And you get 2 FADH2, which are worth 4 ATP. So the total from the Krebs cycle is 2 plus 18 plus 4, which is 24 ATP.
Now the grand total?
8 from glycolysis, plus 6 from the link reaction, plus 24 from the Krebs cycle. That gives us a grand total of 38 ATP.
38 ATP. That is the maximum theoretical yield. Compare that to fermentation, which only gave a net of 2 ATP. Aerobic respiration is 19 times more efficient.
Now, there's a small caveat about that 38 number, right?
Yes. In some animal cells, it costs energy to shuttle the NADH from the cytoplasm into the mitochondria. In those cases, the net yield might be 36 ATP. But for your exams on plant respiration, 38 ATP is the number to remember.
Next concept, amphibolic pathway. For a long time, scientists thought of respiration as purely a breakdown or catabolic process.
That's wrong. It's incomplete. Think about it. If a cell needs to build fatty acids for a new membrane, where does it get the building blocks?
It can pull acetyl-CoA out of the respiratory pathway.
Exactly. And if the cell has excess fatty acids and needs energy, it can break them down into acetyl-CoA and feed them back into the cycle.
The same goes for proteins. Amino acids can enter or leave the cycle at various points.
So respiration involves both breakdown, which is catabolism, and synthesis, which is anabolism. When a pathway does both, we call it amphibolic. Amphi means both.
Last topic for today, respiratory quotient, or RQ.
The definition is simple. It's the volume of CO2 evolved divided by the volume of O2 consumed. RQ equals CO2 over O2.
And this value tells us what kind of fuel the cell is burning.
You have to memorize these values, non-negotiable. First, for carbohydrates like glucose.
The equation was balanced. 6 O2 consumed, 6 CO2 evolved. So 6 divided by 6 is 1.
RQ for carbohydrates is exactly 1.00. Next, fats. Fats have much less oxygen in their structure, so they require a lot more external oxygen to be fully oxidized.
So the denominator O2 is much larger, the RQ will be less than 1.
Yes, for fats, it's typically around source 7. For the example tripalmitin, the RQ is 0.7.
And for proteins?
Also less than 1, approximately 0.9. This covers everything. Glycolysis, fermentation, the link reaction, the Krebs cycle, the electron transport system, oxidative phosphorylation, the amphibolic nature of the pathway, and RQ.
Every enzyme, every step, every calculation. This chapter must now be under your complete control.
There is no margin for error. If you can't remember the link between complex 2 and succinate dehydrogenase, go back and listen again.
If you make a mistake in carbon counting, you will get negative marks. You should be able to draw the entire Krebs cycle on a blank piece of paper in under 60 seconds. Train for it. Today we finished the revision of the most difficult and most important unit. If this revision helped you, like the video and tell us honestly how it was. And if you want more chapters for revision, write the chapter name in the comment section.